Imagine for a second that we don't have to build our next generation of spacecraft in like massive polluting factories.
Right.
Imagine we don't need steel presses or welding torches or these enormous industrial supply
It's
chains.
just hard to even picture.
Exactly. But instead, what if we could just, grow an indestructible spacecraft hull in a giant vat of liquid?
Just brew it.
Yeah, just brew it. And what if the blueprint for that indestructible material came from the exact same protein structure that causes mad cow disease?
It sounds like science fiction.
It totally does. But today, we are completely tearing down everything you think you know about biology. Welcome to the deep dive.
Glad to be here for this one.
So for you listening right now, whether you are trying to catch up on the absolute bleeding edge of biotech or you're just like insanely curious about the future of human evolution, you have found the perfect place to be.
Absolutely.
We are exploring a stack of 3 incredibly detailed, highly speculative, and frankly mind-bending science essays authored by Zachary David Enfield.
Right, from early 2026.
Yeah, early 2026. And the overarching theme we're looking at today is a radical reframing of humanity's relationship with biology itself.
Which is a massive shift.
It is. Think about it. For all of recorded human history, biology has been this fixed, fragile vessel that we just have to endure.
We're just trapped in it.
Exactly. Disease is an invading army. Viruses are these invisible monsters waiting in the dark. Prions are like literal nightmares that destroy the brain.
Yeah, the language we use is so aggressive.
But we are going to flip that script entirely today. We are going to look at how the most terrifying mechanisms in nature are actually the blueprints for the ultimate human technology.
It's a total paradigm shift.
Okay, let's unpack this. Because where do we even begin with a paradigm shift that is this massive?
Well, we really have to begin by completely dismantling and rebuilding our vocabulary.
Okay, makes sense.
The essays argue that we are currently living through the transition from chemical medicine to what Enfield calls information medicine.
Information medicine. I like that.
Right. Because if you think about how we traditionally view illness, our language is entirely built around warfare and physical destruction.
Like fighting a battle.
Exactly. Cancer grows like a weed. Viruses strike like an invading force.
Yeah.
And because our language is physical, our solutions are, well, physical and chemical.
We just nuke it.
Yeah.
Right,
We drop chemical bombs on cancer, basically hoping to poison the tumor just slightly faster than we poison the patient.
Which is pretty brutal when you really think about it.
Total brute force. But beneath every symptom, beneath every malignancy and every infection, lies a much more elegant and, precise truth.
Okay.
Disease isn't a monster. It is simply corrupted code.
Huh.
It's a software glitch running on the most sophisticated computational substrate that has ever evolved anywhere in the universe.
Which is the living human cell.
Exactly. The living, breathing human cell.
I love this framing because it immediately changes the stakes,
It really does.
If a disease is just bad code, like a corrupted loop in a program, then the solution isn't necessarily to drop a chemical bomb on the whole system.
No, not at all.
I mean, you don't take a hammer to your laptop when the web browser crashes, right?
Right, you'd find the bad code and you debug it.
Exactly. You debug it.
That is the perfect way to look at it. If cancer is just a corrupted loop in a cellular division program, the most logical intervention is debugging the code.
Makes total sense.
And if we look at viruses through the same lens of information technology, it changes everything.
Because we usually think of a virus as a destroyer.
We do. But fundamentally, a virus is just an information courier.
Okay.
Its inherent danger doesn't come from the physical vessel itself. The danger is entirely in the payload.
Right. It's the malicious instructions it carries inside.
Exactly. And this is the foundational concept that the author sets up in the first essay. If you strip away that harmful pathogenic payload,
You just take the bad stuff out.
right, and you replace it with constructive code, the virus ceases to be a destroyer. It becomes something entirely different.
It becomes a nanite.
Yes. A microscopic biological machine built specifically for healing and maintenance.
Let's really dig into this idea of viruses as delivery trucks, because for me, this was like the first huge aha moment in the reading.
Brilliant.
A virus, at its core, stripped of the harmful stuff, is just a molecular courier. But what makes it so special is that it isn't just any courier.
No, it's highly socialized.
It has had 3 billion years of relentless evolutionary research and development to figure out how to do its job, with a level of perfection that human engineers can barely comprehend.
What's fascinating here is the sheer brutal efficiency of that evolutionary engineering.
Right.
Human nanotechnologists are only just beginning to grasp the capabilities of a viral capsid.
And the capsid is the outer protein shell of the virus.
Exactly, the outer shell. For billions of years, in every environment on Earth, viruses have been optimizing exactly how to approach a cell.
Like picking a lock.
Literally picking the microscopic locks on the cell membrane, slipping inside without triggering the cell's internal alarm systems, and then navigating straight to the nucleus to deliver their information.
It's incredible.
And they also possess a property called tropism.
Tropism. Okay, what is that?
It's their ability to target very specific tissue types.
Oh, interesting.
Yeah, so some viruses only dock with liver cells,
Ah.
others only target respiratory cells.
So they have an address.
Yes. So when we take a naturally occurring virus and hollow it out, when we literally empty out its pathogenic DNA or RNA, we inherit all of that 3 billion year optimization absolutely for free.
Wow.
We get the ultimate delivery vehicle.
It's like wandering into the desert and finding a highly advanced alien USB drive just lying in the sand.
I love that analogy.
The hardware itself is
Oh,
flawless,
certainly
right?
so.
The casing is indestructible, it plugs into any port perfectly, and the data transfer rate is unbelievable.
Right.
The only problem is that the naturally occurring USB drives out in the wild are loaded with devastating malware.
Tons of malware.
But if we can just plug it into a secure system, delete the malware, and load our own software patch onto it, like a CRISPR gene editing tool, that terrifying virus becomes a microscopic mechanic that we can send into the body to fix things.
Let's take your USB analogy a step further, actually.
Okay?
Because this is not, theoretical physics for the year 2100. This is the reality of clinical medicine today.
It's happening now.
It is.
Yeah.
The essays point specifically to a class of viruses called adeno-associated viruses, or AAVs.
AAVs.
Got it. These are very small, naturally non-pathogenic viruses. They don't cause any known disease in humans.
Oh, so they're safe to begin with.
Exactly, which makes them the perfect blank slate. They have emerged as the leading platform for therapeutic gene delivery.
Wow.
As of the writing of these essays, AAVs are already being used clinically. We're already filling these tiny, perfect protein shells with corrected human code and sending them into the body to replace corrupted sequences.
Right, and the software patch they're usually delivering is CRISPR. Now, I know you listening have probably heard the term CRISPR thrown around in headlines for the last decade, but let's break down exactly what it is in the context of information medicine. It's essentially a molecular text editor, right?
It is the ultimate biological text editor.
Yeah.
CRISPR-Cas9 operates like a search and replace function in a Word document.
Search and replace, okay.
You have a piece of guide RNA, which acts as the search query. It scans the massive, sprawling library of the human genome until it finds the exact sequence of corrupted code.
The typo.
Exactly, the typo. And attached to that guide RNA is the Cas9 protein, which acts like a pair of microscopic scissors.
Okay, so it finds the typo and cuts it out.
All right. Once the guide RNA finds the typo, Cas9 makes a highly targeted cut in the DNA strand.
And then what happens? Does the body just fix it?
Yeah, the cell's own natural repair machinery steps in, sees the break, and patches it using the corrected code that we also delivered in the viral payload.
That is wild.
And the author highlights a massive watershed moment for humanity that happened in December 2023. The FDA approval of a therapy called Casgevy.
I want to pause here because if you listening right now or someone you love has dealt with chronic genetic conditions, this is where the world fundamentally changes.
It really does.
Casgevy was approved for sickle cell disease. For decades, sickle cell has been this agonizing condition where red blood cells deform into these rigid sickle shapes.
It's incredibly painful.
Yeah, they get stuck in blood vessels, causing excruciating pain, organ damage, and shortened lifespans. And traditionally, medicine has only been able to offer painkillers and blood transfusions, just managing the misery.
Right, because traditional medicine was only addressing the downstream physical
The
effects.
symptoms.
Yeah. But sickle cell is, at its root, an information problem.
Okay.
It is caused by a tiny typo in the genetic code that produces hemoglobin, the protein that carries oxygen.
One little typo.
Just one. And Casgevy uses CRISPR to perform a brilliant workaround. Instead of trying to fix the broken adult hemoglobin gene, it targets a different gene called BCL11A.
BCL11A.
Yeah. This is a gene that naturally acts as a brake pedal. Shortly after you were born, BCL11A turns off the production of fetal hemoglobin, which doesn't sickle, and turns on adult hemoglobin.
Oh, I see.
Casgevy goes into the patient's own stem cells, finds the BCL11A gene, and cuts it. It essentially cuts the brake line.
So it turns the fetal hemoglobin back on?
Exactly. The body immediately reverts to producing healthy, flexible fetal hemoglobin. The disease is effectively cured at the source code level.
That is just staggering. In the trials, I remember reading that almost every single patient was completely free of severe pain crises for at least a year after a single treatment.
A single treatment.
You don't manage the symptoms, you patch the code. But sickle cell requires taking stem cells out of the body, editing them in a lab, and putting them back.
Right, ex vivo.
Yeah. The essays mention that the delivery systems are getting even more direct than that, moving directly inside the body.
They are. We are moving toward in vivo editing the code while it's still running inside the living patient.
While the engine is running.
Exactly. Enfield points to phase 1 clinical trials from late 2025 at the Cleveland Clinic for a therapy called CTX310.
Okay, so how does that one work?
Well, instead of a viral shell, this therapy uses lipid nanoparticles.
Like the COVID vaccines.
Yes, very similar. These are essentially microscopic bubbles of fat. The fat bubble protects the fragile CRISPR machinery as it travels through the bloodstream.
Makes sense.
And because it's a lipid, it can seamlessly merge with the lipid membrane of a human cell to drop its payload inside.
And in this specific trial, they were targeting the liver to deal with cholesterol, right? I think I read they were trying to switch off a gene to stop the body from producing so much bad cholesterol.
You've hit on the core mechanism. They were targeting a specific gene that regulates cholesterol production.
Okay.
By delivering a permanent CRISPR edit via these lipid nanoparticles directly to the liver cells, they were able to permanently switch off that specific piece of code. And the results were astounding. They saw a 50% reduction in LDL cholesterol and a 55% reduction in triglycerides in patients who had previously been resistant to all traditional lipid-lowering drugs.
So they just deleted the problem.
Exactly. Think about the implications of that. You don't take a statin pill every day for the rest of your life to chemically suppress your liver's cholesterol production.
And more pills.
You go to a clinic, you receive a single microscopic infusion. It drops permanently.
So what does this all mean for the future of medicine? It means the era of symptom management is closing.
Rapidly closing.
The human body is running on DNA, and DNA is just the densest, most efficient information storage system in the known universe.
It really is.
I saw a statistic in the sources that honestly broke my brain for a minute. It said that theoretically, 1 single gram of DNA can store over 200 petabytes of data.
It's hard to even conceptualize.
Right. Just to visualize that for you listening, 200 petabytes is roughly equivalent to every single digital movie, photo, and song humanity has ever recorded, all fitting into a single drop of liquid that you could balance on the tip of your finger.
The informational density is beyond anything we can build with silicon. And this brings us to the profound realization that ties the first essay together.
Which is?
Because DNA is a universal language, the exact same biological alphabet of A, C, T, and G is used by every single living organism on Earth. Understanding it as an information medium opens a door that has been locked for human history.
Ah, because it's universal.
Exactly. If we can use viral couriers to patch broken human code, then the next logical step is to look around us. We aren't the only ones running code.
Everything is running code.
Every plant, every insect, every animal on this planet is running a highly optimized software program, and we can read their code.
Which brings us perfectly into the second part of the essays, borrowing from nature's library.
Yes.
This is where we cross the boundary from simply healing human disease to actual human augmentation.
The upgrade phase.
And I have to admit, reading this section, my mind was racing. Because if you think about it, evolution has already solved almost every environmental problem humanity faces.
It has.
We worry about extreme cold, extreme radiation, lack of oxygen, massive physical trauma. But somewhere out there in the wild, some animal has already beaten those problems.
Right. They have the software patch.
We just didn't know how to download it until now.
The author introduces a framework he calls functional translation.
Okay, functional translation.
In the past, if we wanted the properties of an animal, we had to wear its skin or harvest its organs.
Which is, yeah, pretty primitive.
But we don't need to physically transplant animal tissue into a human being anymore. We just need to read the biological solution that is encoded in their DNA.
Just copy the code.
Exactly. Once we understand the molecular mechanism, the exact sequence of proteins that solves the problem, we can use synthetic biology to rewrite a human-compatible version of that exact sequence. We translate the animal software into human code.
Let's talk about the ultimate survivor in the animal kingdom, because the essays spend a lot of time on this. The tardigrade,
Oh, the water bears.
the microscopic water bears. For anyone who hasn't seen a picture of these things, they look like tiny, eight-legged vacuum cleaner bag bears.
That is an oddly accurate description.
And they are practically indestructible. They have been frozen at absolute zero, they've been boiled, they've been exposed to the vacuum of space. But most incredibly, they can survive levels of ionizing radiation that would turn human DNA into absolute confetti.
The radiation resistance of the tardigrade is one of the most remarkable biological anomalies we've ever studied.
How does it even work?
Well, when human cells are hit by high-dose ionizing radiation, like X-rays or gamma rays, the energy rips through the water in our cells, creating highly reactive molecules called hydroxyl radicals.
Okay.
These radicals act like microscopic buzzsaws, shredding the physical strands of our DNA, causing massive mutation, cell death, and radiation sickness.
Yeah.
But the tardigrade has evolved a specific software patch for this. They produce a unique protein called DSUP, which stands for damage suppressor.
And how exactly does DSUP stop the buzzsaws? Because radiation isn't something you can just put a physical shield up against at the microscopic level, right? It penetrates.
It's actually a brilliant piece of molecular architecture. Inside the nucleus of a cell, DNA is wrapped around protein spools called nucleosomes, which helps pack the massive length of DNA into a tiny space.
Okay, like winding a hose on a reel.
Right. Now, the DSA protein is positively charged and DNA is negatively charged. The DSA protein physically binds to these nucleosomes, wrapping itself around the DNA like a microscopic, flexible suit of armor.
Oh, wow. So it literally coats the DNA.
Exactly. When the radiation hits the cell and creates those hydroxyl radical buzzsaws, the DSA protein acts as a physical barrier, absorbing the damage and physically shielding the DNA strand from being shredded.
That is amazing.
And the truly incredible part is that this mechanism isn't limited to tardigrade biology. It translates.
Wait, it translates to humans.
Yes. When human cells in a lab are genetically engineered to express the tardigrade DSUP protein, their tolerance to lethal X-ray radiation increases by roughly 40%.
But wait, I have to stop you there. Are we talking about permanently mutating human DNA to make us radiation-proof? Because I don't want my permanent genetic code altered with tardigrade DNA just so I can get a safer dental X-ray.
No, of course not.
It seems like the risks of permanent modification would outweigh the benefits. Isn't there a temporary way to do this?
You are bringing up the exact safety concern that researchers flagged immediately, and this is where the convergence of technologies becomes so important.
Okay.
The MIT research from early 2025 that the essays cite took a much more elegant approach. They did not permanently alter the human genome.
Thank goodness.
Instead, they used the lipid nanoparticle technology we just discussed. They packaged synthetic mRNA messenger RNA that contained the instructions for building the DSA protein.
Oh, so just a temporary message.
Precisely. They delivered this mRNA into human cells. The cells received the message, manufactured the tardigrade armor for a few hours, and then, as mRNA naturally does, the instructions degraded and disappeared. The cell stopped making the armor.
If we connect this to the bigger picture, the practical, everyday applications are just unbelievable.
Huge.
Imagine a cancer patient who needs aggressive radiation therapy. The biggest problem with radiation is that it destroys the healthy tissue surrounding the tumor.
Right, the collateral damage.
But what if, an hour before the treatment, you give the patient a localized injection of this DSUP mRNA targeted at their healthy tissue?
It protects them.
The healthy cells temporarily build tardigrade armor, the tumor remains exposed, you blast the area with radiation, the tumor dies, and a few hours later, the patient's healthy tissue clears the mRNA and goes completely back to normal.
Exactly. Or think about astronauts heading to Mars. They get a solar flare warning, they inject a dose of DSUP, their bodies are shielded for the duration of the storm, and then they return to normal.
It is the pinnacle of functional translation. We are borrowing transient survival software from the extremes of the natural world.
And Enfield's essays don't stop at extreme environments. They look at physical performance too.
Like what?
For example, deep-diving mammals like seals and whales. A Weddell seal can hold its breath and dive for over an hour.
An hour?
How? It's an information solution. Their muscles contain concentrations of myoglobin, the protein that stores oxygen in muscle tissue. that are up to 30 times higher than what is found in humans.
30 times. Wow.
By delivering a targeted viral nanite carrying optimized human-compatible regulatory sequences that ramp up myoglobin production, we could drastically increase human oxygen storage capacity.
Okay, I have to push back hard here. We're crossing a line.
You think so?
We are moving from protecting a cancer patient to creating literal X-Men.
Mm.
If we are talking about splicing in seal code so a human can hold their breath underwater for an hour, or giving people temporary tardigrade superpowers so they can walk through a nuclear reactor, this feels like a massive, massive ethical leap.
It is a profound leap.
The line between curing a disease and creating a superhuman seems completely blurred here.
And the author dedicates the final portion of his work entirely to the ethical architecture required to manage this, which we will definitely get into. But in the immediate term, Enfield grounds these augmentations in medical necessity.
Okay, how so?
Think about the myoglobin upgrade not as a scuba diving superpower, but as a lifeline. For a patient suffering from severe chronic obstructive pulmonary disease, COPD, or late-stage emphysema, every breath is a struggle.
Right, their blood oxygen is so low.
Exactly. Their blood oxygen levels are critically low. If you could increase the oxygen storage capacity of their tissues by even a fraction of what a seal can do, you give them their life back. You take them off the oxygen tank.
Oh, wow. I didn't think of it that way.
It also has undeniable applications for military personnel operating at extreme high altitudes where oxygen is scarce. Remember, synthetic biology means we aren't creating human-animal hybrids. We're writing tightly controlled, human-optimized code that is merely inspired by the animal's molecular solution.
And what's really haunting about this section of the reading is that sometimes we don't even need to borrow the code from an animal.
Right.
Sometimes the solution is already inside our own DNA, just sitting there in the dark, turned off. I'm talking about the salamander regeneration studies. That blew my mind.
The axolotl is the classic biological marvel in this space. It is the only known tetrapod that retains the ability to regenerate complete, fully functional limbs throughout its entire adult life.
So if it loses a leg?
If an axolotl loses a leg, it doesn't just form a scar. The cells at the site of the amputation actually reverse their aging process.
They reverse it.
Yeah, they de-differentiate back into stem cells, forming a mass called a blastema. And from that blastema, they regrow the bone, the muscle, the blood vessels, and the incredibly complex nerve networks, perfectly integrated with the rest of the body.
And we are starting to figure out exactly what commands trigger that process. The essays mention a 2025 study out of Harvard that mapped the trigger to the sympathetic nervous system. The physical injury actually creates a body-wide chemical memory, driven by the adrenaline pathway, that primes the animal stem cells to wake up and start building. But the absolute kicker for me was the research out of LSU from 2024.
This is where it gets crazy.
They suggest that humans aren't actually missing the genetic blueprints to grow back an arm or a leg.
That is the paradigm-shattering realization. The genetic toolkit for complex tissue regeneration isn't alien to us. We use those exact same tools to build our bodies in the womb.
Right.
The genes for limb formation are already in our DNA. But the regulatory elements that activate these genes, specifically molecular pathways known as FGF and have been evolutionarily suppressed in adult humans.
Suppressed? Why would we turn that off?
Well, at some point in our evolutionary history, the survival benefit of quickly forming a thick scar to stop bleeding and prevent infection outweighed the immense energy and time required to slowly regrow a perfect limb.
Oh, that makes sense. If you're bleeding out, you just need a quick patch.
Exactly. So, evolution put a padlock on those genes. What the LSU research indicates is that a targeted viral nanite wouldn't need to deliver foreign salamander DNA into a human wound.
It wouldn't.
No,
Yeah.
it would only need to deliver the specific transcription factors, the molecular keys, to flip our own dormant embryonic switches back into the on position.
We don't need to build a new machine. We just need to find the lost passwords to the machine we already are.
Exactly.
It's honestly breathtaking to think about. We can upgrade our biological software, we can patch genetic diseases at the source pin, we can temporarily wrap ourselves in radiation armor, and we can potentially convince our bodies to regrow lost limbs just by waking up ancient code.
It's a lot to process.
But everything we've talked about so far is just the software. What if we could take biology and use it to radically upgrade our physical infrastructure?
Moving to the hardware.
What if we used biology's most indestructible hardware to build our world? Here's where it gets really interesting, because we have to talk about the prion paradox.
The prion paradox. This is the moment in the essays where the narrative takes a sharp, brilliant turn. We leave the realm of personalized medicine and enter the realm of material science, civilizational infrastructure, and structural engineering.
Now, when most people hear the word prion, if they know it at all, they think of mad cow disease or Creutzfeldt-Jakob disease.
Right. Highly fatal diseases.
They think of untreatable, 100% fatal neurodegenerative diseases. From what I understand, a prion is basically a misfolded protein that acts like a microscopic zombie.
That's a good way to describe it.
It bumps into healthy proteins in your brain and physically corrupts them, forcing them to fold into its own broken shape, leaving the brain looking like a sponge. It is the stuff of absolute medical nightmares.
Pathologically speaking, you are entirely correct. Mammalian prion diseases are terrifying because they subvert the body's own proteins. But Enfield asks us to strip away the pathology and look strictly at the architecture.
Just the physical structure.
Exactly. The physical structure that prions and functional amyloids share is known as the cross-beta architecture. And from a purely structural engineering standpoint, the cross-beta architecture is a technological marvel that human engineering cannot match.
Why is it so special?
It consists of alternating layers of incredibly rigid, hydrogen-bonded structures called beta sheets. These rigid sheets are separated by flexible protein side chains that act like microscopic shock absorbers.
So it's rigid but flexible.
Yes, you have extreme rigidity paired with energy-dissipating flexibility at the molecular level.
It's a crazy mental pivot. It's like discovering that a highly toxic substance, say asbestos or radioactive waste, is actually the single most perfect material for building spacecraft and skyscrapers. As long as we can figure out a safe engineered version of it.
Right.
Because when you look at the mechanical properties of these protein structures, the numbers are completely absurd.
Absurd is exactly the right word. When material scientists began engineering polymeric amyloid fibers based on this architecture, the results shattered existing paradigms.
Boy, how much?
Well, these engineered biological fibers demonstrate a toughness of 160 megajoules per cubic meter.
I don't know what that means in practical terms.
To put that in perspective, natural spider dragline silk has long been the absolute gold standard, the holy grail of biological supermaterials for its tensile strength.
Okay.
Engineered prion fibers beat spider silk.
Wow.
And they achieve this incredible toughness at roughly 1/8 the density of steel. You have a material stronger than spider silk, lighter than carbon fiber, and built entirely out of protein.
And they are practically indestructible. That's what makes them so hard to kill in a medical setting, but so incredible for engineering. The essays note that these prion fibers survive being boiled in concentrated pure formic acid.
Yes.
That is a condition that would instantly melt, denature, and destroy any normal protein or biological tissue.
Yeah.
And they are highly resistant to gamma ray radiation that would completely fry traditional silicon electronics or weaken traditional metals.
This extreme resilience brings us to a fundamental engineering concept called hydrogen bond density, or HBD.
Okay.
Enfield describes this as the volume knob for materials engineers. In a cross-beta structure, the core is held together by hydrogen bonds. If an engineer designs the amino acid sequence to increase the hydrogen bond density in the core of the fibril, the material becomes incredibly stiff, rigid, and strong, similar to structural steel or titanium.
So you just turn up the knob to make it stiffer.
Exactly. If they decrease the hydrogen bond density, the nail becomes highly dynamic, flexible, or fragile. This means that using one single underlying biological technology, an engineer can tune the physical properties across a massive spectrum just by tweaking the sequence of the protein code.
But the absolute craziest part of this isn't how strong or adaptable they are, it's how you manufacture them.
The brewing process.
Right. If I want to build a titanium hull for a submarine, I need a massive, energy-intensive factory. I need mining, smelting, thousands of degrees of heat, and massive physical presses. But you don't build prion fibers in a factory, they build themselves.
It's even stranger than that. You don't build them, you brew them. It is known as the seeding paradigm.
Seeding paradigm.
Imagine a massive vat of liquid. Inside is a harmless liquid protein monomer, just a soup of raw protein building blocks floating randomly.
Just floating there.
You take a microscopic fragment of a preformed prion fiber, this is the seed, and you drop it into the vat. The moment that seed touches the liquid protein, it templates it.
Templates.
It physically forces the surrounding liquid proteins to fold into its exact rigid structure, adding to its length. As the fiber grows, the gentle mechanical mixing of the vat causes the fiber to snap.
And then the broken pieces.
Each broken piece becomes a new seed, which starts growing its own fiber. It is an exponential self-replicating chain reaction. It requires absolutely no energy input, no extreme heat, no toxic chemical catalysts. You're literally brewing structural supermaterials at room temperature.
And I want to make sure we emphasize a crucial point that Infield is very careful to clarify. We are not using the mammalian prions that cause mad cow disease.
Oh, absolutely not. That would be insane.
Right. No one is brewing vats of fatal brain diseases. The engineers are using yeast prions, like one called SUP35, which are completely 100% harmless to humans.
Yes.
They have no biological mechanism to interact with human proteins. Or they are using entirely synthetic proteins designed from scratch. So what you have is a completely safe, self-growing, indestructible material that beats the strength of spider silk and costs basically nothing to manufacture compared to industrial metals.
Well, applications are endless.
You could build aerospace composites out of this. You could build deep-sea submarine hulls that withstand crushing pressures.
And let's look at the maintenance of those structures. Because the material grows from a seed, it retains the ability to self-repair.
Wait, self-repair?
Yes. Imagine a deep space exploratory vessel with a hull made of an engineered amyloid composite. During its journey, it gets struck by a micrometeorite, creating a microfracture in the hull.
In a traditional ship, that's a huge problem.
That's a structural compromise that requires a physical repair. But in a biological hull, the exposed broken ends of the prion fibers react with a dormant, protein-rich liquid matrix embedded within the hull's layers.
Oh, wow.
The broken end acts as a new seed, and the crack literally heals itself by rapidly growing new fiber across the gap until the structural integrity is restored. We are talking about biological armor that heals like human skin but is stronger than steel.
Okay, so prions can build indestructible, self-healing spaceship hulls and bulletproof vests in a vat of liquid. But the sources go somewhere else entirely.
They do.
They say these proteins can also build computers. This is the section of the essays where I really had to pause, get up, walk around, and reread the pages. The author calls it the computational soup.
The computational soup is where the two halves of our discussion, DNA as the ultimate software and prions as the ultimate hardware, finally converge.
Okay, let's break this down.
It is the proposition of fusing DNA data storage with prion conductivity to create a biological supercomputer. We've already established that DNA is incredibly dense, capable of holding petabytes of data, but it also has a mathematical advantage over our current technology.
Right?
Standard silicon computing is binary. It relies on microscopic transistors being either on or off, representing zeros and ones. DNA operates in 4 distinct chemical states: adenine, cytosine, thymine, and guanine, A, C, T, and G.
So it's quaternary.
It is a quaternary computing medium. Fundamentally, it can carry double the information per physical operation compared to a binary system.
Right, I understand that DNA is an amazing hard drive. It can hold a ton of data in a tiny space. But if DNA is the hard drive, I'm trying to picture how we actually get the data in and out fast enough to use it as a computer.
That's the challenge.
If I plug a USB drive into my laptop, I know there are electrical signals flashing in nanoseconds reading the memory. But DNA is a biological molecule floating in a liquid vat. Usually when we talk about biological processes, we're talking about chemical reactions.
Enzymes.
Yeah, enzymes drifting over, attaching to a strand, slowly pulling it apart to read it. Chemical reactions are incredibly slow on a computational scale. You can't run an AI supercomputer if you have to wait for an enzyme to chemically process a piece of data over the course of an hour.
You've hit on the exact bottleneck that bioengineers have been wrestling with for decades. If you rely on chemistry to read and write your data, DNA computing is essentially useless for anything requiring real-time speed.
So how do they speed it up?
This is where the essays introduce a radical physical workaround: terahertz radiation.
Terahertz radiation. Okay.
The terahertz gap is a band of electromagnetic frequencies sitting between microwaves and infrared light. Now, think about how a microwave oven works. It blasts water molecules with radiation, causing them to spin wildly, crash into each other, and generate massive amounts of heat.
Which would cook the DNA.
That destroys delicate biological molecules.
Yeah.
But terahertz radiation is different. It doesn't cook the molecule. DNA has natural vibrational frequencies that sit exactly in the terahertz range.
Oh, is this like the opera singer analogy? Where if the singer hits the exact right pitch, the precise resonant frequency of a wine glass, the glass vibrates and shatters without anyone touching it.
That is the perfect analogy. The hydrogen bonds that hold the two strands of the DNA double helix together are like the wine glass. By hitting the DNA with incredibly short, intense pulses of terahertz radiation, tuned to that exact resonant frequency, You can physically shake those hydrogen bonds apart.
You literally shake it open.
You unzip the double helix physically, read or write the data using optical sensors, and then let it rezip as it cools, all within a matter of picoseconds.
Trillionths of a second.
Yes. You completely bypass the slow chemical enzyme reactions and use resonant physical frequencies to achieve read-write speeds that rival or exceed silicon processors.
Okay, that makes sense for reading the hard drive. So instead of a microchip inside my laptop, I have a microscopic vat of DNA zapped by terahertz lasers to read the data. But how does that vat actually interface with our macro world? If I want to plug a monitor into this biological soup or send an electrical signal to a keyboard, how do I connect a physical copper wire to a single microscopic strand of DNA floating in liquid?
That interface problem has been the absolute bane of biological computing. Trying to connect a traditional silicon or copper wire to a biological molecule is, as the saying goes, like trying to plug a fire hose into a teacup.
Oh, wow. Yeah, the scale is totally off.
The scale mismatch is insurmountable. Even our smallest silicon traces are massive, clunky monoliths compared to the elegance of a single DNA molecule. If you try to run an electrical current from a metal wire into DNA, you usually just fry the biological material.
Right.
But remember our indestructible yeast proteins, the prion fibers.
Yes, the super strong material we're using for spaceship hulls.
Exactly. It turns out strength isn't their only property. Prion fibers can actually conduct electricity.
Hey, really?
They possess a quantum mechanical property called tyrosine hopping. Embedded deep within the core of the prion fiber are amino acids called tyrosines, which feature aromatic carbon rings.
Okay.
When the protein folds into its rigid beta sheet structure, these tyrosine rings align perfectly, spaced at exact, precise distances. They act like stepping stones across a river. An electron can hop from one tyrosine ring to the next, traveling rapidly down the length of the biological fiber.
So they are actual wires.
They act as naturally growing, highly efficient nanowires. And the crucial detail, the piece that makes the whole architecture possible, is their physical size. Prion fibers naturally grow at a diameter of roughly 10 nanometers.
Which is, wait, that's the same scale as the DNA, isn't it?
Precisely. There is no scale mismatch. The prion nanowires grow themselves at the exact same sub-nanometer scale as the DNA molecules. In the computational soup, the DNA molecules act as the massive quaternary storage nodes. The terahertz lasers act as the ultra-fast clock speed, reading and writing the data. And the self-assembling prion fibers act as the physical interconnects, the logic gates, and the electrical wiring, linking billions of DNA nodes together in a massive three-dimensional web.
It's a whole system.
It is a unified biological computer operating entirely at a scale that silicon manufacturing can never reach.
That is genuinely mind-bending. A supercomputer that literally grows its own wiring inside a jar. But there is a glaring, massive problem here that I keep coming back to.
What's that?
We talked about the seeding paradigm. You drop a prion seed into a vat of protein and it just grows. It replicates exponentially in all directions until it uses up all the liquid.
Right.
How do you stop a self-replicating supermaterial from just growing into a giant, useless, tangled blob of biological wire? If we are talking about engineering, if I want to build a specific logic circuit, or a highly structured bone scaffold, or a spaceship hull with specific dimensions, how do I tell the liquid protein to grow here but not there?
You've identified the chaos inherent in self-assembly. Uncontrolled growth is useless for engineering. And that exact problem brings us to the third essay and the breakthrough concept that ties everything we've discussed today into a controllable, programmable reality, prion optogenetic lithography.
Prion optogenetic lithography. That is a mouthful. In my notes, I just wrote down painting with light.
Painting with light is the most accurate way to visualize it. This technology is the brilliant synthesis of three completely different scientific disciplines.
Okay, what are they?
First, you have the prion self-assembly mechanism, which acts as our physical building material, our ink. Second, you have photolithography. This is the use of highly structured light to write microscopic patterns, which is traditionally how we etch circuits onto silicon wafers in the semiconductor industry.
Right, printing with light.
And third, the magic ingredient, optogenetics.
Let's break down optogenetics for the listener because it sounds like science fiction, but it's very real.
Optogenetics is a biological technique that involves engineering proteins so that they physically change their shape or their behavior in response to very specific wavelengths of light.
Oh, like plants.
Yes, nature already does this. Plants have light-responsive protein domains, like Cry2 or LOV2, that allow a sunflower to track the sun across the sky. Scientists have learned how to isolate these light-sensitive domains and fuse them to other, completely unrelated proteins to act as an on-off switch.
And the essay highlights a very specific recent breakthrough here from 2025, the PHOBIT system developed at Texas A&M University.
Yes. PHOBIT stands for Photo-Inducible Binary Interaction Tool. It's essentially a modular, incredibly precise light switch for proteins. Here is how they solved the blob problem.
Okay, how?
They took a harmless yeast prion monomer, the building block that wants to self-assemble, and they genetically fused the PHOBIT light switch onto it. The result is a modified protein molecule that is completely inert and dormant as long as it is in the dark.
So it won't grow.
You can have a massive vat filled with these proteins, and they will never assemble. They just float there harmlessly in the liquid.
But the absolute second you shine a specific wavelength of light onto the vat.
The switch flips. The PHOBIT domain absorbs the photon, physically changes its shape, unlocks the prion monomer, and suddenly that specific protein molecule becomes capable of templating and growing into a rigid fiber.
That is so cool.
But the key is this. The assembly only happens within the exact microscopic volume of liquid that the light is actively touching. Everywhere else in the vat, the liquid remains dormant.
So it's like a 3D printer, but there is no nozzle moving back and forth and no mechanical parts. You just shine a complex 3D holographic light pattern, like a literal hologram, into a completely still pool of liquid protein.
Exactly.
And instantly, a solid, complex object crystallizes and grows out of the liquid, but only in the exact shape of the light.
The resolution is limited only by the wavelength of the light, which means it operates at the molecular scale. You can shine a microscopic light pattern into the computational soup to instantly print a vast, perfectly structured 3D electrical circuit.
Wow.
But the essays go further. You can use it to print biological structures. One of the more colorful examples Enfield provides is printing a vegan steak.
A steak.
Yeah. You fill a vat with nutrient, liquid, and muscle-forming proteins attached to these light switches. You shine a complex holographic pattern into the vat that perfectly mimics the intricate micrometer-scale marbling and fiber texture of a Wagyu ribeye.
No way.
The proteins assemble instantly along the light paths. You pull out a steak that is structurally indistinguishable from animal muscle, grown in seconds using light.
Okay, the holographic Wagyu steak application is wild, but let's pull this back to human medicine because the implications for healing are what really shook me.
They truly are staggering. Imagine a trauma scenario. A patient comes into the emergency room with a severely shattered femur. The bone is crushed into pieces too small to pin together.
Usually that's an amputation.
Right, or massive metal implants. Instead, a doctor injects a specialized liquid solution directly into the shattered leg. This solution contains dormant, light-sensitive proteins designed to form an ultra-strong osteoconductive scaffold.
Basically a temporary, highly porous artificial bone.
Yes, that human cells love to grow on. The liquid flows into every microscopic crack and gap of the injury, completely harmless and liquid.
And then the doctor brings out the light.
Yes. The doctor uses a device that emits a highly focused, computerized array of near-infrared light from outside the body. Near-infrared light is unique because it safely penetrates deep through human tissue without causing damage.
Oh, so they don't even have to open the leg back up.
Exactly. The doctor projects a 3D hologram of a perfect, healthy femur straight through the patient's skin and muscle into the liquid pool inside the leg.
And the proteins react.
Wherever that holographic light intersects with the liquid, the proteins instantly snap together, assembling into a rigid, permanent, highly structured, therapeutic scaffold that exactly bridges the shattered bone.
That is unbelievable.
The liquid in the dark areas simply flushes out of the body naturally. Over the next few weeks, the patient's own bone cells use that scaffold to perfectly regenerate the femur.
So you could literally 3D print a regenerating bone matrix inside someone's body without ever making an incision. Or, going back to cancer, you could inject the liquid, shine the light exactly on the tumor, and instantly print a toxic microscopic net that solidifies around the tumor and physically strangles its blood supply.
Yes, you can.
You are writing physical, life-saving architecture directly into living human tissue. And the programming language you're using is just light.
Exactly. The holographic light defines the precise geometry, and the prion cascade instantly fills that geometry with physical mass. It is the ultimate convergence of biology, optics, and material engineering.
Okay, let's take a very deep breath here, because we need to look at the totality of what we are discussing.
Thanks a lot.
We are talking about humanity gaining the power to literally 3D print biology inside the human body using light. We are talking about rewriting the fundamental source code of human DNA.
Right.
We are talking about turning our fragile cells into radiation-proof supercells by borrowing from tardigrades and building self-repairing supercomputers out of the proteins that cause brain diseases.
It sounds like magic.
The potential for human flourishing is unbelievable, but the potential for catastrophic disaster here is equally staggering.
This raises an incredibly important question, and it is a testament to the author that he does not shy away from the darker implications.
Which we have to talk about.
The entire sixth part of his first essay is dedicated strictly to the architecture of programmable evolution, the immense risks involved, and the absolute non-negotiable necessity of built-in safeguards.
Because we are crossing a fundamental threshold. For the last 4 billion years, evolution has been a blind, random process.
It's a very slow process.
It relies on random mutations, massive amounts of trial and error, and usually millions of years to adapt to an environment. But with these technologies, we are moving to intentional, programmable evolution. We are taking our hands off the passenger door and grabbing the steering wheel of our own biology.
And with that control comes profound technical and existential risks. On a purely technical level, there is the ever-present risk of runaway expression.
Like the blob we talked about.
Exactly. We talked about how prions replicate exponentially. What happens if a self-replicating biological nanite or a bone scaffold we are printing inside a patient somehow forgets to turn off? What if the off switch mutates?
Right. Cancer, fundamentally, is just runaway, uncontrolled cell growth. If we are intentionally putting rapidly self-replicating nanites or structural proteins into the human body, a slight coding error could accidentally trigger massive synthetic tumors. The bone scaffold just keeps growing until it crushes the leg.
Which is precisely why Enfield insists that biological kill switches cannot be an afterthought or a regulatory add-on. They must be a foundational architectural requirement for any synthetic biology.
So how do they work?
The essays detail a mandate for a two-level kill switch system for any internal application. First, you have a genetic molecular off switch.
Okay.
The synthetic code of the nanite includes a promoter region that requires the constant presence of a specific, rare, small molecule drug to stay active. It's a dead man switch.
Oh, like a pill you have to take.
Exactly. If you stop taking the pill, the nanite instantly loses the chemical signal required to function, and it degrades.
That handles the software. But what about the hardware? If a prion-based scaffold has already built a massive physical structure inside the body, turning off the software doesn't make the indestructible protein block disappear.
That requires the second level, the structural dissolution switch. If the physical protein scaffold goes rogue or simply finishes its job and needs to be cleared, the architecture includes a backdoor vulnerability.
A built-in weakness.
Yes. Doctors can introduce a highly specific engineered enzyme or a CRISPR-edited chaperone protein that acts as a targeted solvent. This solvent is designed to break down only the specific hydrogen bonds of the synthetic structure,
And that's it.
melting it harmlessly back into liquid amino acids that the body's kidneys can filter out without harming a single natural human protein.
Okay, so it seems the technical safeguards are theoretically robust. But I want to step back and look at the human element, the societal element, because the socioeconomic implications of this tech terrify me more than runaway bone growth.
It's a very valid fear.
Let's go back to Casgevy, the CRISPR therapy that functionally cures sickle cell disease today. The essays mention that the current cost for that single treatment is roughly $2.2 million per patient.
It's prohibitively expensive for most of the world.
Now project that forward. If this technology eventually allows the ultra-wealthy to literally upgrade their DNA, to buy tardigrade radiation resistance, to buy seal-level oxygen capacity, to buy enhanced cellular regeneration or cognitive processing speed, we aren't just looking at traditional income inequality anymore.
No, we're not.
We are looking at a permanent biological class divide. We are talking about humanity literally physically splitting into two distinct species: the genetically enhanced elite and the unenhanced baseline population.
The author issues a stark, uncompromising warning about exactly this scenario. He states that the governance of synthetic biology and programmable evolution must be treated as a global commons problem immediately right now, before commercial interests and private monopolies have locked the technology behind insurmountable paywalls.
It has to be accessible.
If this technology is allowed to create a biological class divide, it will be far more profound, entrenched, and irreversible than any existing pharmaceutical inequity or economic disparity in human history.
But how do you even begin to govern something that powerful? You can't just put a blanket ban on it because you'd be banning the cure for sickle cell and cystic fibrosis.
You start by drawing very strict, globally recognized ethical lines regarding the target of the intervention. Enfield explains the absolute critical distinction that must be maintained between somatic enhancements and germline enhancements.
Somatic and germline, can you break those down?
Somatic modifications affect only the body cells of the specific consenting adult who is receiving the treatment. If you choose to get a transient mRNA injection to protect your healthy tissue from radiation, that changes your body.
Right.
But those changes are isolated to you. They die with you. They do not enter your reproductive cells, which means they do not get passed down to your children.
But germline modifications.
Germline enhancements modify the DNA within human embryos, sperm, or eggs. Those changes become a permanent part of the human gene pool. They are passed down to the patient's children, their grandchildren, and every subsequent generation forever.
That's a huge line to cross.
The author references the massive international scientific condemnation of He-Jian Kui in 2018, the biophysicist who shocked the world by editing the CCR5 gene in human embryos to confer HIV resistance. Germline editing means making permanent, irreversible biological and evolutionary decisions on behalf of countless future descendants who have absolutely no ability to consent.
It's playing God with the entire species.
Enfield argues forcefully that while we should rapidly pursue somatic medicine to heal the living, germline modifications must remain under the strictest possible international moratorium. We can patch the adult hardware we currently occupy, but we do not rewrite the ancestral lineage of the human species until our philosophical and ethical frameworks are vastly more mature than they are today.
So what does this all mean? We have covered an immense, staggering amount of ground in this deep dive.
You really have.
We've traveled from viewing the human body as this fragile, fixed vessel that we just have to suffer in, to viewing it as a remarkably resilient, fully customizable computational platform.
A completely new way of seeing ourselves.
We've redefined viruses from these invisible killers into microscopic couriers capable of delivering life-saving code. We've taken prions, the absolute stuff of neurological nightmares, and transformed them into the ultimate cosmic building material, self-assembling computers, and healing scaffolds written into existence by holograms of light.
It is a total, irreversible paradigm shift in how we understand life. The body ceases to be a fixed vessel governed by the blind luck of natural evolution. It becomes a platform as customizable as software, yet as resilient and adaptable as life itself. We are learning to speak the source code of nature.
It is a massive amount to process for you listening. It changes how you look at a virus, a disease, or even your own reflection. But I know you have one final thought, one last provocation from the essays for us to ponder as we wrap up today's journey.
I do, and it steps entirely outside of our current century.
Oh, I like the sound of that.
If we connect everything we've discussed today to the vast, cosmic, bigger picture, think about the distant future.
Okay.
If humanity eventually masters this synthetic biology, if we truly learn to build our deep space exploratory spacecraft, our planetary supercomputers, and our very civilizational infrastructure out of genetically programmed, self-repairing, DNA-based proteins.
If we just build our world out of life.
Yes. And if we journey out into the cosmos enclosed in these living biological ships, what happens if that technology outlives us by 1,000,000 years?
Oh, wow.
If another intelligent civilization out in the stars someday stumbles upon a self-repairing, self-contained, DNA-based biological structure floating in the void of space, will they even recognize it as advanced technology?
Or will they just think it's an alien animal?
Exactly. Will they scan it, see the proteins in the DNA, and just assume it's a naturally occurring spacefaring organism? It makes you wonder, as we engineer life to become our ultimate technology, are we just closing an ancient loop on what life actually is in the universe?
Oh. Wow. That is the kind of question that is going to keep me awake staring at the ceiling tonight. A biological ship indistinguishable from life. Thank you to everyone listening for joining us on this incredibly expansive deep dive into the future of our biology. Keep questioning the world around you, keep looking beneath the surface of what you think you know, and we will see you next time.